Hi, welcome back to analyze the single cell RNA-seq data using the scanpy tool. So in the previous video, we have seen how we can download the data from 10x website, 10x Genomics website, how to create an AND data object and run the QC steps on the AND data object, and run all the basic analysis like performing PCA, dimensionality reduction, finding the neighbors, and UMAP visualization. So we are going to see the downstream steps from the part where we have left off. The only difference between the Jupyter Notebook from part 1 tutorial and this video is that I have created an AND data object. I have saved the AND data object, the expression values in adata.raw. That's one difference. And the second difference is that I have not copied the AND data object to a new AND data object, which I've done in the previous video. So I would be running all the analysis on the A data. So that's the only difference instead of creating a new copy. So with that said, let's move on to the downstream analysis. So all these steps we have seen, finding neighbors, so you can refer to my previous video. So in this video, what we are going to do is uh, we are going to look at the downstream analysis where we are trying to look at the um, Leiden algorithm to cluster the cells that we have seen in the previous step. Also, we want to look at the differentially expressed genes and identify the marker genes for each cluster, and we want to annotate each of the clusters. So why do we need to cluster the cells? Basically, we want to identify the cell types. For example, this cluster of the cells may contain B cells, T cells, natural killer cells. So we want to identify the clusters and what are differentially expressed genes in the specific cell type. Also, we want to look at subpopulations of the cell. For example, if it is T cell, is it CD4 T cell or CD8? So we want to further cluster into a distinct subpopulation and want to find them. We also want to know the cell state, like when the cell is active, what are the specific genes which are expressed constantly. So we want to find the marker genes, and we want to use those marker genes to look at the biological pathways. Also, what makes the difference between the cell type A and cell type B is some of the genes are expressed in cell type A and some in cell type B. So there must have definitely been an overlapping process between the two cells. So we want to see how cell type A has become a cell type B. So basically we want to infer the trajectory of cell state A to cell state B using the expression data analysis. So previously we have seen in the UMAP plot, we basically see three clusters. So we want to do the cluster much better using the algorithms available in scanpy tool. So basically we load the additional packages. So you can see we have our AND data object, and I am running here this tool and Leiden algorithm on the AND data object. So you can see I now have a unstructured object where it is Leiden here. So in this function, I have not given the resolution. The default resolution is 1. So let's see what resolution means. So Resolution is actually a parameter which controls the coarseness of the clustering. So do I need to have more clusters? I may need to increase the resolution. If I want to look at the abstract level of all the cell types, I may have a lower resolution. Then the question comes, what's the optimal resolution value? So unfortunately, there is no specific value recommended. It depends on the data set that the person analyzing the data set. So basically, we can start from a lower resolution and slowly increase the resolution. And whichever fits um, and one feels sensible, he can use that resolution as a input parameter for the function. So basically what I'm doing here is trying to find the cluster count for each of the resolutions. So basically I'm creating two different lists. One is the cluster counts, another is resolution. So I'm going to loop through the resolutions and trying to plot and trying to find out the number of clusters. So basically when I run this and see, I am getting a list of cluster counts, and I have the resolutions here. So when I plot the resolutions versus the number of clusters here using the matplotlib function, so I can see as my resolution increases, I see the number of clusters also increasing. So that's one thing. So the next step, so basically we might be choosing our resolution to be 0.5 here. That's because in the tutorial I see 8 different clusters being used, so possibly it fits this resolution. So I'm just going through what the tutorial has been used, that the parameter that tutorial has used. This is an additional parameters which can be used also when you are running the Leiden algorithm, which is called key added. So by default it will be, the resolution will be 1, so Leiden underscore 1 key is added. If I want to change my resolution and I want to include that in my AND-data object, I can do that as well. Simultaneously, I can have multiple resolution uh, run on the AND-data object and store the cluster information in the same AND-data object under different columns. So that's what I'm doing here. So I'm running the clustering algorithm on at different resolutions, and I'm adding a key inside the AND data object. So I save this, I run this, and I'm going to plot this. All I need to do is color, and I give all the keys that I've provided here: Leiden underscore 1.0, 1.0, 0.6, 0.4, and 1.4, and I give how many columns I want. So by running this, I can see like for 0.4 resolution, I have something like 6 clusters. 0.6, I have 9 clusters, so on. This is just for my own sake. I just wanted to see how it looks like when I loop it. I want to see for all the resolution, instead of manually adding one by one here, what I wanted to do is from certain resolution to another X resolution to Y resolution, I wanted to see the plots of all the uh, 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 cell clusters. So that's what I am doing here. So from resolution 0.1 to 2, increase stepwise 0.1, run the clustering algorithm, increase the resolution every time by I, and I add the key here. So that would create a column for each of the resolution that I'm um, asking in the loop. So which is, you can see in the ops data frame, you can see for each um, resolution I have a column with the cluster number in the column. So this is basically a small for loop to create a list. So okay, basically I need to run this for loop, then I would have a data object, and then I run this filter. Now I have this list of all the column names. Now I pass the name of the column names as a list here to this plotting tool. So this would now create um, UMAP plots for different resolutions, and you can see number of columns is 2. I have 2 columns, so 0.6, 0.4, 0.1, and okay, this is because we have previously added this. That's why these keys are also being here. But you can see from 0.1, 0.2, 0.3, 0.5, 7. So you can see all these keys at different clusters at different resolution. Basically, this is just for an exploration sake, but as I said previously, we will stick with resolution 0.5. So I am using the 0.5 here. So this gives me 8 different clusters. So if I want to embellish a bit more, I can add an outline. Outline is true. Also, I can have a legend location on the data. So here you can see the legend on the right, but when I have this, I have the legend on the cells. So let's run this. So you can see all my clusters. I have only the cluster numbers here on top of the cells here, cell types different. So at any point, if you want to look at the count of the cells for each cluster, we can run this value counts on this specific column. So it would give you the counts of the cells for each cluster here. Okay, now we will move. Now we have the clusters here. Now we are good to find the differentially expressed genes between these clusters. So basically, I was trying to read the scanpy paper. It looks like the term marker genes and the differentially expressed genes are interchangeably used. You can see identification of marker genes for clusters via differential expression tests. So we will be annotating the clusters by identifying the marker genes between the clusters. So for this, we would be using the rank underscore genes underscore groups function, which basically uses two different kinds of tests, t-test and Wilcoxon test. This would actually generate as a plot. So for cluster 0, what are the genes, differentially expressed marker genes? So this is top 25 here. We are seeing we have specified top 25 here, and y-axis you would see the score. So the higher the score, higher it was the expression, and lower p-value, which means it's very significant. And the lower score means lower expression, possibly higher p-value. So for all the 8 clusters, we can see the genes which are highly expressed, and so at this point, I want to bring to the attention that when running this function, trying to find the differentially expressed genes, this function would use the ANDData.raw that we have previously created in the part 1 of the uh, document. Um, okay, so we run the t-test. So that would generate us the plots. Then to understand a bit more, t-test is actually a parametric test, and it relies on the means of the groups and calculates the p-value. So uh, there is a chance that uh, it is it can be affected by the outliers, and it's a bit more sensitive than the Wilcoxon test. So we have seen the posts, people and researchers using the Wilcox test because it's a rank sum test, and median is used. It's less sensitive to outliers than compared to t-tests, but we will compare both in our tutorial. So we would run the same function with the Wilcoxon test, and we are going to add a key added here. So same thing, we will plot the top 25 genes, and as you see here, I'm printing both the AND data object as well as um, the number of genes used for the Wilcoxon test, or finding the differentially expressed genes. You can see the AND data object is made up of 1826 genes, which we have quality filtered and selected highly variable genes. We performed all our PCA and dimensional reduction, but we use the differentially expressed genes. For differential expression analysis, we use all the genes. This would give us a better idea of uh, expression, gene expression, and the significant genes. So I've just created a data frame. So this data frame, we can see these are the cluster names, the cluster numbers, and for each cluster number, this is the top gene. LDHP is the top gene here, and with the RPS 22nd in the second one. And you can see in the cluster 0, LDHP is the top gene, RP27 is the second top gene. So similarly, for each of the clusters, we have created a table here. So we basically have to choose the genes from this table. These are the marker genes, and we do the cluster annotation. But before we do that, I just want to show you this creating a violin plot. So basically what I've done is I've taken the top gene for each of the cluster, and I've plotted here. So you can see LDHB. So I will zoom it. Looks like this LDHB gene is present in quite a few clusters, but predominantly it looks like much expression has been in cluster 0. Also S100A9 looks like the second cluster, definitely. That's why it's more predominant in the second cluster. That's why it's on the top. Why similarly the CD79A is the top gene in the cluster 2. So we can see here it's predominant. It has more predominant expression in this cluster 3. So you can see similarly like the other clusters. Then we would like to save these differentially expressed genes, with all this information into a file. So basically what we can do is we can take all the 32,000 genes, and this piece of code would generate the gene in the cluster, fold and its score and its log fold change, p-value and adjusted p-value. Similarly, in the cluster 1, the top best gene, score, fold changes, p-value, and so similarly, all the 8 groups would have the same information. So we can use this data frame actually to filter for the p-value, log4 changes, and finally coning down to a smaller data set where we are interested in. So just for the comparison, we will see the class in the cluster 0. We'll compare the genes, how many genes t-test um, uh, has found, and Wilcox has um, identified as a significantly expressed gene. So we use the p-value cutoff and log2 fold change, same but for both of them, both of the statistical tests, and we save the sets here, and we import this function matplot libven, and we draw the Venn diagram for both the data sets for gene sets. So we can see the Wilcox test seems to be more conservative, less affected by the outliers, whereas the t-test, which is in my understanding is affected by the mean, so looks like it has found more number of uh, um, differentially expressed genes. So now we move on to the differentially expressed genes, how we can visualize them. Basically, we have all the data in A data. We want to see the top 5 genes, and we have um, we have used the Leiden 0.5 resolution, and we use the t-test 0.5, and we want to show the gene labels. So we provide these parameters into the heat map function, and we plot. This would create a heat map plot with all the genes, top 5 genes for each cluster. You can see here, and the expression value, and it also provides the dendrogram. The same thing can be plotted as a dot plot, almost same parameters. We just run this plot, just that we change the dot plot heat map to dot plot. Rest of the parameters almost remain the same. That would generate as the dot plot. We can see the expression is high for this gene, specific gene is high in cluster 1, whereas not much in the rest of the clusters. This is stacked violin plot, same parameters, only the function changed. So yeah, someone who wants to visualize in a different way. This is almost same as heat map, just that it shows the matrix plot, almost same parameters, actually same parameters, and we can visualize the genes which have higher expression. So basically we choose the genes from these visualization plots, and also based on the prior information, we make a marker list, and we are interested in looking at the expression plots for this marker genes. Ideally, we may have to do that, but in this case, since we already had the prior information of from the tutorial, what genes are highly expressed, so we just go through the same list that tutorial has provided. So we generate a list of marker genes, and we assign manually the cluster names for each of the clusters that we have produced. Um, and then we plot the uh, clusters. So another way to plot is, as I've shown previously, we add the outline and uh, also legend location on the data, so you can see the information, the plot like this. So similarly, we can draw for the set of marker genes we have created in the list. Also like this, the dot plot and the matrix plot. But the only difference is that previously we had only the cluster numbers here, but now we have the cluster names because we have annotated them. So in summary, we have learned how to cluster using the Leiden algorithm, Leiden using the scanpy tool. We have identified the differentially expressed genes for each cluster. We have annotated the clusters, and we have visualized the differentially expressed genes or markers of interest, marker genes of interest. So see you in the next tutorial. Thank you.